I'm Mariane Martinez, and my thesis advisor is Dr. Dan Harrington, and I work in the Farach-Carson Harrington lab. So my research project involves studying epithelial cells in 3D culture systems. So typically, cells have been grown in traditional 2D systems, and as this may work for cell lines, primary epithelial cells are a little bit more hard to grow. So using a 3D hyaluronic acid culture system allows us to grow epithelial cells and allows them to retain a more in vivo morphology and phenotype. So my research actually has involved working with prostate cancer cells, and these primary prostate cancer cells have been previously grown in a mouse xenograft model, and these mouse xenograft models are really hard to maintain. They're very expensive, so that has kind of pushed us towards developing an in vitro system in which we can grow these cells. And our studies have actually shown that a HA or hyaluronic acid hydrogel seems very promising towards growing these cells in vitro and may allow more easily manipulation of these cells when compared to mouse xenograft models. I think directly a broad application of this would be to develop an in vitro bone metastatic drug screening model of these prostate cancer cells. But I think even more broader, this system can be applied to different cell types, and that includes tissue engineering or other primary cells that you want to grow that won't grow in 2D. So I think this is applicable to many cell types and can allow better growth and better representation of your cells in vitro. So prostate cancer is actually the second leading cause of cancer death in American men. And most of those cases are actually from metastasis to bone. And unfortunately, there's no effective treatment for the metastatic disease of prostate cancer. So our cells are actually taken from the bone tissue of a man and have been maintained in a mouse xenograft model. So they retain expression and morphology of these primary cells and allow for a better system of drug screening. The best part of my doctoral research experience has been interacting with other students that are as passionate about science as I am. And not only passionate about science, but many of these are also passionate about community outreach programs. So that has opened doors for me to be able to use science and to be involved in my community all at the same time. So.